Welcome back to biochemistry class, and now we will discuss about gene regulation. Gene regulation is basically about how certain genes are expressed and how others are not. Gene regulation is the process of controlling which genes in a cell's DNA are expressed or used to make a functional product such a protein. So different cells in a multicellular organisms may express very different sets of genes even though they contain the same DNA. So dito sa gene regulation, mayroon tayong tinatawag na dominant and recessive na traits or alleles, and the gene for that alleles are sometimes not expressed and sometimes are expressed. The set of genes expressed in a cell determines the set of proteins and functional RNA it contains, giving it a unique properties. So yung set of genes daw na na-express determines kung ano yung proteins na mabubuo and yung functional RNA na taglay nito. So, nagkakaroon ng unique properties because of that gene regulation kung ano yung gene na ma-express or yung mato-turn on, turn off. In eukaryotes like humans, genes expression involves many steps, and gene regulation can occur at any of these steps. However, many of these genes are regulated primarily at the level of transcription. So yung transcription, di ba, nangyayari dun is yung from the coding strand of DNA, and it was paired by the complementary RNA polymerase. So dun sa part na yun, nagkakaroon na agad ng gene regulation mostly sa mga eukaryotes. Gene regulation is how a cell controls which genes out of the many genes in its genome are turned on or expressed. And thanks to gene regulation, each cell type in your body has a different set of active genes, despite the fact that almost all cells of your body contain the exact same DNA. So, di ba, meron lang naman tayong exact DNA, but paano, di ba, from a cell, from a single cell, from our mother and father, nagkaroon ng combination, and how that certain cell nagkaroon ng iba't ibang functions to our bodies. Di ba? We have the brain cells, we have the blood cells. So, paano sila nagkaroon ng pagkakaiba-iba knowing that we just only have parang certain DNA, and that is because of the gene regulation. Meron lang na mga traits or meron lang na genes na i-express sa isang cells, and meron din na tago. So, kaya nagkakaroon tayo ng uniqueness. Yung cell na yun nagkakaroon ng uniqueness. These different patterns of gene expressions cause your various cell types to have different sets of proteins, making each cell type uniquely specialized to do its job. So, your parang ibibigay kong additional task sa inyo is to identify kung ano-ano yung mga proteins na within our bodies and to identify yung function nila. So, aasahan ko yan. So, for example, one of the jobs of the liver is to remove toxic substances like alcohol from the bloodstream. To do this, liver cells express genes encoding subunit of an enzyme called alcohol dehydrogenase. So, it is a amino acid enzyme. It is an enzyme kasi nagtatapos sa ASE. Pag nagtatapos sa ASE, pakakatandaan natin na is itong enzyme which somehow contains protein. So this enzyme breaks alcohol down into a non-toxic molecules. The cells of the liver don't send signals using neurotransmitters, which is from our brain. Hindi daw galing sa hindi yun naging hindi yung neurotransmitter yung naging signals for that. So they keep neurotransmitter genes turned off. Yung neurotransmitter is not turned off, but yung alcohol dehydrogenase yung turn on na gene for that. So many copies of a protein can be made from one mRNA molecule. This is because several ribosomes can translate the same mRNA molecule at the same time. This cluster of ribosomes that translate a single mRNA sequence are called polyribosomes or polysomes. But how do cells decide which genes turn on? Turn on. We can say that cells' gene expression pattern is determined by information from both inside and outside of the cells. So it depends sa mga instances na nangyayari inside and outside the cell. Examples of information from inside the cell is that the proteins it inherited from its mother cell, whether its DNA is damaged, and how much ATP it has. So, yung ATP is adenosine triphosphate. So, nangyayari daw yung inside the cell na parang yung information is nadidetermine ng information if in the instances like mother cell daw where DNA is damaged. So, nagkakaroon ng parang regulation because of that happenings inside the cell. Example naman of information from outside the cell, when a chemical signals from other cells, mechanical signals from the extracellular matrix, and nutrient levels. So from another cells, nagkakaroon ng signals na makakaapekto to decide on which genes will be turned on. As an example, let's consider how cells respond to growth factors. A growth factor is a chemical signal from a neighboring cell that instructs a target cell to grow and divide. We could say that the cell notices the growth factor and decides to divide. So, yung example na ito is yung cell mismo, yung naka-notices that there is a need for the growth, need for growth. So, na-activate yung growth factor na yun. And that is an example of how do cells decide which genes will be turned on. So remember that cells don't make decisions in the sense that you or I would. Instead, they have a molecular pathways na sinusunod that convert information such as the binding of a chemical signal to its receptor into a change in gene expression. So yung chemical signal na yun to its receptor, yung magsisilbing parang command na magkaroon ng pagkakaiba sa pag-express ng genes or yung gene expression. Here is the example. The cell detects the growth factor, kung makikita nyo sa figure, through physical binding of the growth factor to a receptor protein on the cell surface. So, from the growth factor to the receptor. So, binding the growth factor causes the receptor to change shape. So, yung receptor nag-change shape siya for the purpose of triggering a series of chemical events in the cell that activate proteins called transcription factors. So, because of that change, parang pag-change ng shape ng receptor, nag-trigger siya for a series of chemical events. The transcription factors bind to the certain sequences of DNA in the nucleus and cause transcription of cell division-related genes. So, nandito na yun. Division promoting gene, growth promoting gene. And the products of these genes are various types of proteins that make cell divide, drive cell growth, and push the cell forward in the cell type. So, ito naman, response element versus enhancer. So, we will differentiate response element versus the enhancer. Response elements are short sequence of DNA within a gene promoter or enhancer region that are able to bind specific transcription factors and regulate transcription of genes. Under condition of stress, the transcription activator protein binds to the response elements and stimulates the transcription. So yun na nga, yung response element is usually a short sequence of DNA lang na pwedeng mahanap natin or we can found at the enhancer region. While the enhancers, so it is also a short regulatory elements of accessible DNA that help establish the transcriptional program of cells by increasing the transcription of target genes. So, dito sa enhancer, nag-a-act siya, that element acts for increasing. So, from the word kasing enhance, mas pwede natin siyang i-relate for increasing. So, that's all for my report. So, my co-reporters will discuss to you about mutation and the other subtopics. Thank you for the listening.